अलाउ करती हूँ इसको, ठीक है। So hello everyone. It's 5 p.m. So we'll start with the problem solving session. I hope you can hear me clearly. If there is some audio issue, please let me know. So I'm Mochandra Paul. I'm a PhD scholar in the Department of Chemical Engineering at IIT Kanpur, and I'm also a PMRF fellow. So in this during this lecture, we'll discuss few problems based on the content that is being taught, that has already been taught in week 1. So I hope you have gone through the week 1 content yourself and is already familiar with the basics of molecular biology and like genetics, the basics of the genetics. So if you are clear with those prerequisites, you will be able to solve the problems. And if you understand the problems that I'm solving today, you can definitely solve the current assignment that is given for week 1. So starting with the first problem, uh, I'm reading it. You can also read and try it out yourself. Adenine forms a complementary pair with blank in RNA and blank in DNA. Okay, so we have to fill in the blanks here basically with one of the 4 options. For understanding this problem, you should recall the base pairing rule. So what is the base pairing? Yes?
And the option will be C.
Yes, the option will be C. You are correct. According to the base pairing rule, in RNA, the adenine will form 2 hydrogen bonds with uracil, and in DNA, it will adenine will form 2 hydrogen bond with thymine. You are correct. Moving on to the next question. Here we are given a sequence that is present in a newly synthesized RNA. This is the sequence that we are given. Okay, and then we have to identify among these 4 options which is the correct sequence for the DNA template. So you can take a pen and paper and try to figure it out. I'm also doing it so that you can match your steps. So here it is RNA, right? So for DNA, the detection option A. So wait a second, a few more people are joining. So hello all. So those who have joined late, let me introduce myself briefly. I'm Ochandra, and today I'm going to solve few problems based on week 1 content. Okay, so previous problem you guys have missed. Let me just quickly recall. Here the question says what is what is the base pairing happen in DNA and RNA. Okay, so have a quick read. I think it's quite easy. Everyone can understand this. This question follows the base pairing rule, simple base pairing rule. In RNA, adenine forms 2 the hydrogen bonds with uracil, where in DNA it is with thymine. DNA and RNA, it's easy. Okay. The next question is this. Others who have just joined now, please have a look at this question. Okay, so here the sequence is given for the RNA. Here this is it. We have to determine what will be the sequence for the DNA template. One of you have already answered, but let me just work it out for those who haven't able to figure it out. Okay, so if we again, one person is joining. So here in the DNA, the sequence will be this. T, right? Then in place of there will be A, G, G, T, C. Again, another person is joining. People are becoming late. No problem at all. Okay. And in place of A, there will be T. Correct? All right. So I think I've not done it correctly. I think so. Let me go back. I'm a little bit nervous since this is my first time teaching here. Okay. So no problem. We can do it, right? So A, base pairs with T, right? U will be A, G, G, then will be T. No, here will be A, correct? A. In D, this is for DNA. Okay. For G, there will be C. C. For T, there will be A. Correct. So if we see the 5' direction, the answer will be this, right? No. Answer will not be A then. The answer will be B. Did everyone understand? I think someone answered A, I think. Could you cross check and figure it out? Let me know if it is clear to all. If it is clear, the answer will be B. Okay, then I'll move on. Okay. So we have to first write the sequence in 3 to 5' direction for the DNA, as this is will be the template, right? But in the options, we have to then read from the 5' direction. So when we read in the 5' direction, T A C C A G G A T. Okay, so the correct answer will be B. The next question is very easy. Which of the following cells from the same individual would have the difference chromosome number? So this question is based on the understanding of cell division, right? For mitosis, the chromosome number in the daughter cells remains the same, whereas in meiosis, the chromosome number become half, right, in the daughter cells. So which of these options do you think will be correct? So in the same individual, the liver cells, neurons, white blood cells, these are what? Somatic cells, right? They are involved in normal physiological functions, whereas this is produced in the gonads, right? In female. So, and this is the cell that is involved in reproduction, right? During reproduction. So, and this is a haploid cell. So here the chromosome number will be 23 instead of 46. The correct answer will be A. Moving on to the next question, it's also easy. So the question says that, okay, wait, another person is joining. So in this question, they're asking, like, you can solve this question if you are able to understand the structure of the chromosome. And then you can easily fill in the blanks. So in the condensed form, when the chromosome is present at the metaphase stage, you can identify there are regions, right? Centromere regions to which the spindle fibers connects, and there are telomeres, the two extreme ends of the chromosomes. So, so using that knowledge, you can fill in the blanks here, correct? The region of the chromosome that joins a pair of sister chromatid is known as the two sister chromatids, which are exact copies. They are joined at the centromere, correct? So A has the centromere first, D has the centromere, and blank the chromosome ends are referred to as the extreme ends. They referred to as the telomere. So the correct answer will be D. If anyone has any query, they can unmute themselves and we can discuss. Okay. So this question reads, the central dogma of molecular biology is an explanation of the flow of genetic information within a biological system. Now, based on your knowledge of the processes involved in the cellular compartment where the processes take place, choose the option that identifies the correct pair. So you guys are all familiar with the concept of central dogma, right? This is universal. The DNA, the genetic information in the nucleus, is like made into copies, right, in form of RNA. So the process of transcription. So the genes in the entire DNA sequence, only the gene part forms the mRNA, right? The transcription process involved, and this RNA, the sequence of the RNA, are present in form of genetic codons. These genetic codons are then translated to form protein, the sequence of amino acids. So this basic knowledge and also the location. So transcription or then replication, this all happens in the nucleus, whereas the mRNA, which is formed in the nucleus, is transported out into the cytoplasm where the actual translation process happens. So using this knowledge, you can definitely understand what will be the answer. Can anyone like reply? Like, what do you think is the answer of this question? So based on your knowledge of the processes involved and the cellular compartment where the processes take place, choose the option that identifies the correct pair.
Ma'am, I think it will be option B.
Option B. What about others?
Option D, translation.
Okay. So if someone is saying B, someone is saying D. So let's work it out. Here the replication, it happens in the nucleus, right? So based on the process, it happens in the nucleus, the replication. And replication, it doesn't involve copying the DNA into RNA. So this option will go off. Then transcription, it happens in the nucleus, right? And so this is already wrong. So this B will not be the answer. But transcription happened in the nucleus, and then but it doesn't involve copying RNA into protein. This is what happens in translation. So C is wrong. But this translation, it happens where? It happens in the cytoplasm, right? So there are tRNA, rRNA, the ribosomes, all present in the cytoplasm. And translation is the process where RNA is copied into protein. So the answer will be D. Okay. So moving on to the next question. An essential and unessential component are excised off from the pre-mRNA strand with the help of splicing factors to generate the mature mRNA. Now identify the component which is not part of the mature mRNA strand that must be translated. So this question is based on the concept of splicing that happens just after transcription. So during transcription, we get a premature mRNA which have the coding and the non-coding regions, right? We term them as like introns and exons. So exons are the part which are actually retained in the mature mRNA. Introns are removed. Okay. And this is the splicing. The splicing process make it happen that it removes the intron, selectively cuts the double-stranded DNA at the two regions to remove the intron sequence and then joins the two adjacent exons. This is the splicing. And apart from splicing, what happens in the 5' end, they add like a cap is added, and the 3' end a poly A tail is added. Okay, so here they are asking among these four options which is not part of the mRNA strand. Okay, so exon must be there. Okay, poly A tail is added at the 3' end, and 5' UTR, it is also an exon actually. So it is the intron which is removed. So the answer will be A. Okay. If anyone have any query, they can unmute themselves and ask. Okay. I'll now move on to the next question. Now, during this process of splicing, which of the following is not required? Okay. Uh, we need like the following options. Have a look. We need pre-mRNA molecules, right? It is the molecules that is just formed after the transcription process. What about the ribosomes? The ribosomes are not involved in transcription, right? They are involved in translation. Then there is spliceosomes, and also there is small nuclear ribonucleoproteins. So spliceosomes are actually the main complex which is actually doing the splicing process, and it consists of small nuclear ribonucleoproteins. Okay, so during splicing we need mRNA, like pre-mRNA molecules. We need the spliceosome actually contains the small nuclear ribonucleoproteins, but ribosomes are not needed. So the correct answer here will be ribosomes. If anyone have any query on this question, they can ask. It's fine. So then I'll move on to the next question. If anyone like is joining late and like have only heard the half of the question and they want me to repeat any following question, they can ask me. Okay, so that I can go back and again they can have a look. By the way, this meeting is being recorded so that you guys can get it later. So this is the 8th question. It says protein isoforms are the proteins which are often coded by the same gene, comparable to each other, and perform similar roles inside a eukaryotic cell. Now, which of the following processes lead to the generation of such protein isoforms? So, is everyone familiar with the term protein isoforms, right? So, what is protein isoform? So, there is one gene, okay, and due to any one of this process, we get different forms of the protein that is generated from that particular gene only. And these different variants of the protein, they perform similar function. So what is the process? They want to know what is the process that is leading to the formation of such variants of the same kind of a protein. Okay, so the answer here will be alternative splicing. So what happens in alternative splicing? Uh, this is, say, these are the introns and these are the exons. This is a premature mRNA. Okay, so this is exon 1, 2, 3. So while splicing, what can happen? 1 and 3 are joined together in the mature mRNA, or 2 or 3 can join together. Okay, so in this, our 1 and 2 can join together. So we get 3 different kinds of mature mRNA. And when these 3 different kind of mature mRNAs get translated, they form 3 different types of proteins, which comes from the same gene. And this mRNA is actually transcribed means, from the same gene in the DNA. So this is the alternative splicing. Okay, this alternative splicing process gives rise to protein isoforms. Simple enough, right? If anyone have any queries, please let me know. Now moving on to the next question. Here what they're saying that there is an mRNA strand and it consists of 120 nucleotides. And upon translation, this mRNA will form a polypeptide chain. So they are asking like what will be the length of this polypeptide chain, and also we have to assume that the stop codon will code for one amino acid. So we are aware that there are things like if we divide the sequence of the mRNA into 3-letter words, we get one genetic code, right? Genetic codon. So this is 120 nucleotides can be divided into this is A, B, C. If these are the nucleotides, so we can divide into 3 such words sequentially. So we can just simply, what we can do to get the number of amino acids, and each such codon codes for one amino acids. Simply just 120 divided by 3, right? That will give me the what total number of amino acids. It is 40 amino acids, correct? So the correct answer will be 40 amino acids. So the whatever polypeptide chain that we'll get will have 40 amino acid. Could everyone understand this? It's quite easy actually. Okay. Now moving on to the next question then. Now for solving this question, let me give you guys like website which you can refer in the chat box. Okay, I'll share in the chat box. So if you have received the message, okay. So this question for this question, we will need to refer to the list of the amino acids, okay, and also the 3-letter word which is used for each amino acid, like glutamine is written as GLN, and also the 1-letter word. Okay, so this website will help you get the entire list, as well as you will need to know how many codons code for a particular amino acid. So using that information, you can easily solve this question. So have a look, okay, and try it yourself. Meanwhile, I'll also try. So a newly synthesized polypeptide chain, it has the following sequence. So this is the 1-letter code for the amino acid. So M stands for methionine, L is for leucine, I is for isoleucine, and Y is for tyrosine. Okay, so you have to then look up for the codons of each of these amino acids. Okay, now which one of the following options identifies the correct sequence in the mature mRNA that is used to form this polypeptide chain? So after, so you have to first get the sequence of codons, okay, corresponding to each amino acid. All right, and then you have, so in the website they'll give for the DNA, you have to convert for RNA, simply replace thymine with for uracil. Okay, and then write what is the sequence. The answer will be that only. Okay, I request you all just to try it out. It's very easy actually. Just you need a pen and paper to do it. Okay, and remember that the codons are degenerate. Okay, so one amino acid can be coded by multiple codons. So keeping that in mind, you have to solve this question. I request you all to just try it out. So if you go to the website, which the link for which I've given in the chat box, you can check the following. Okay, the AUG, the first. Let's do one option at a time. AUG here, that you will find that it codes for the amino acid methionine. Okay. Then for leucine, L stands for leucine. Now it is coded by two codons actually, TTA and TTG. Now for mRNA, you have to consider U in place of T. Okay, so in this option, you have UUA. Okay, here also you have UUA. Okay, but in this option, the first option is here also the first option is AUG. So I feel like B, A and D, one of them might be correct. Okay, then for isoleucine, you can check the codon for isoleucine. The three-letter code for isoleucine is ILE. It is coming from three codons, ATC and ATA. Okay, then again you have to replace T with U. So here you can see AUU. So far, these two options look similar, right? So methionine, leucine, isoleucine. Then again there is leucine. All right. So leucine can be coded by another codon, UUG, right? So here I can see UUG, but here then it goes off. Correct. Here it is not UUG. Then again, if you go for but I feel like this is the correct answer, but again let's check the sequence for tyrosine as well. The sequence for tyrosine is TAT, correct, and TAC. So in place of T you can replace U. So this is the, so yeah, so my hunch was right, A was the correct answer. And yes, you have to have a stop codon, and the stop codon will not come in your sequence. Okay, so a protein sequence, the polypeptide sequence, there the stop codon will not come, but it's give the signal that here the synthesis has to be stopped, the protein synthesis has to be stopped. So the correct answer is A here. Now moving on to the next question. An allele is what? Like, is it a mutant version of a gene, a variant of a gene that may not cause a disease, or one of the several variants of a gene present in normal population, or all of these? So like I would request, like, if one of you can speak like what they understand by mutant, sorry, what they understand by the term allele. This question is based on the definition of an allele. Yeah.
Partner or something like that.
Yeah.
Allele means they are always come in a pair. So allele is like one of the partner of a pair.
Okay. Okay. So you're partially correct. You're partially correct. Would anyone like to try? Anyone else would like to try? So you can. So so there is for the chromosome, a single chromosome doesn't exist. There's a pair. Chromosome exists in pairs, right? This is called homologous chromosome. Correct. So this is one gene which is present in one chromosome, say A, right? And some variation, like caused by a point mutation or frame shift or anything, because of which a variant is created of this particular gene, and it is present in one of the homologous chromosome. Okay, so this is one variant. Now this variant is present in one person, one individual. Now for this particular gene A, if there is mutation in some other location of this particular gene, another variant is created, and that might be present in another person. Okay, so different individuals can have, a few variations, like non-lethal variations of this same particular gene, and each of these variants or each of these mutants will be called allele. Okay, so allele is nothing but a gene where, due to mutation, a variant is created, and this variation is non-lethal, means it is not affecting the survival of the person. So option A is correct in this case. Okay, then option B, as I said, it should not affect the survival of the person. It shouldn't cause difficulty in living, like lifestyle should not be hampered, like the skin color, right? People are of different kinds of skin color. Few are white, few are like brownish, few are like darkish shades. The different shades are there, but they're living a normal life, they don't have a disease. So allele is also that it shouldn't create any disease. All right, it shouldn't result into a disease. And then, as I said, for one particular gene, various kind of variants can be present, and different individual can have different variants. So it is then allele is then one of the several variants, like you can call it allele 1, allele 2, allele 3, and whoever having the normal gene will be that person will be called the wild type, and others will be called the mutants. Okay, so the option C in this case is also correct. So the final answer will be D. So all of these options actually refer to the definition, like explains what is an allele. Okay, if anyone have any query, they can ask me. Now moving on to the next question. They're asking, like, where would you find two allelic genes? I already explained in my previous question that they are present in two homologous chromosomes, right? So it's you cannot find allele of particular gene in another homologous chromosome, like in chromosome number 22, which is having two homologous chromosome. The gene which is present in one of the chromosome of the 22 will be present only on the homologous pair. Okay, that is another pair of another copy of the chromosome 22 that is coming from one of the parent, right? You can understand like where the homologous chromosomes are coming from, right? So one is coming from the mother, another coming from the father. So here, in this case, the answer is option B. Is everyone clear on this question? If anyone have any doubt in understanding what is homologous or non-homologous chromosomes. Right. Now the next question. Which of the following changes in the DNA will most likely lead to a frame shift mutation? So what do you mean by frame shift mutation? Would anyone like to pitch in on this? What do you mean by frame shift mutation? Like, it is one of the types of the frame shift mutation. You must have heard somewhere, like in class 12th also they teach it.
Ma'am,
Yeah.
basically a frame shift mutation is one where we can see that there is an addition or an deletion of a base pair in the DNA.
Okay. So because of addition or deletion, what happens?
It is not a multiple of three. It is something insertion and deletion, but it is never of multiple of three. That's why it's called as a frame shift.
Actually, I couldn't understand. Can you repeat the answer that you said? Ajay, is it?
Frame shift is insertion or deletion of a chromosome, but it is not in a multiple of three. It's gene.
So I think you are wrong. Means yes, there will be. Uh, Hello, Mansi. You have raised your hand. You can ask if anything you have. Yeah.
Mutation which causes change in codon reading triplet.
Yes. So your means all of you, like all three of you, have given partial answers to the correct thing. So what happens is due to addition of one or multiple bases or deletion of one or multiple bases, the reading means how the protein synthesis machinery will read the mRNA, okay, or how the RNA making machinery, okay, we will read the DNA. It is going to be changed. It is going to be changed, okay. So let me give an example. Say this is your sequence, okay. This is your sequence, and I have here at this place inserted a base pair here. Now, initially I was reading like this, 3 at a time. Okay. Now my reading will be 3, then 3, then 3, and like this. So because of this addition, and here ATG was coding for one amino acid, it was coding for another amino acid, correct? So, but now ATG is same, but since due to the addition of A here, now, the amino acid which was previously supposed to be there because of triple T, now that will be replaced by the amino acid which is coded by A double T. Okay. Then here also the amino acid which was coded by CCT is now replaced by TCC. So in this way, the sequence of amino acid is also getting changed, and the original amino acid sequence is not at all retained with addition. And if we delete it, the same thing will happen again. So the reading, how the machinery which is going to read the DNA or the RNA sequence, the is being changed. And here actually it is called open reading frame. Here 3 which I'm counting, each is called the open reading frame. How I'm reading the sequence. So here you have what they're saying ki, which one of this mutation, missense mutation, nonsense mutation, or insertion, or all of these, which one will lead to this frame shift? Means the way I'm reading the sequence is being changed. Okay, so as per this example, you feel it is C. Okay, uh, what about nonsense mutation? Does everyone understand nonsense mutation, right? So what happens in nonsense mutation? It's a point mutation, means one single base pair is replaced, but it is replaced in such a way that a stop codon comes instead. So say this is a sequence. यहाँ पे मैं डाल देती हूँ T T, okay, sorry, T18. Now if the base is changed here, TAG. So TAG is a stop codon. Okay, TAG is a stop codon. Then TTG, TAA. These are like few of the stop codons. Now here, now the base pair changes such that this particular codon is converted into one of the stop codon. Then a nonsense mutation is happened, and it will lead to a smaller polypeptide chain. So here frame shift is not happening. What about missense? Missense, but here the change in one base pair is leading to another amino acid, like what it happens during sickle cell anemia. In sickle cell anemia, that is a condition where one base pair A is changed to T, because of which when A was present, the codon, resulting codon was coding for glutamic acid, but now because of T, it was coding for valine. So the correct answer will be insertion, right? So because of insertion, we are getting a shift in the open reading frame, and the resulting amino acids after that is being changed. So the answer is C. Okay. So the next question is again based on the different types of mutation. Here it's saying which one of the following mutation type is not likely to generate a truncated protein. So as I told in previous case, nonsense mutation, right? Because of nonsense mutation, we get a stop codon at a place where we are not supposed to have, right? It was present somewhere downstream, but we're getting the stop codon upstream before the entire sequence could be completely translated. So nonsense mutation is giving then a smaller protein, a polypeptide chain, which may not be functional anymore. So this nonsense protein is resulting into a truncated protein. Then there is frame shift mutation, right? So in frame shift mutation, as I told you, what is happening. So what do you then missense mutation, and then lastly there is point mutation. So which one of these would you think is not likely to generate a truncated protein? Point mutation is just a permanent change in a base pair in the DNA sequence. Okay. Should anyone answer? Yeah.
Frame shift.
Frame shift.
Frame
okay.
shift.
Frame shift. Okay. Okay. What about others?
Yeah, it would be missense mutation.
Missense. Okay. Would anyone else would like to try?
Point mutation, madam, it will generate a protein, but not a truncated protein.
Okay. Anyone else? So point mutation, what happening here? You're getting, you're just replacing, right? A replacement in the base pair is happening. And this point mutation is seen in missense mutation as well as in nonsense mutation, because one base pair is being changed. It's a type of mutation which explains the result of missense and nonsense mutation, if you think like that. Okay. But so nonsense mutation will definitely result in truncated protein, so it is definitely not the answer. Neither is the point mutation, because as I told you, it is what is happening at the DNA level. But when you think point mutations manifestation at the protein level, means you get a stop codon because of which the protein is truncated, or you get a change in amino acid. So the amino acid in the final polypeptide chain is being replaced. Then we get the missense. And what frame shift? The frame shift, it can lead to truncated. Think like that, means like how? Because of the frame shift, if you add or delete, you can get a premature stop codon. If you delete some sequences, and when you then see the DNA sequence and then make three sets, you can get a stop codon early. So truncated truncated protein can result from frame shift mutation, but from missense, it is not like that, right? Missense uh, means that the resulting amino acid in the protein, okay, is being is different from what was the original one. So here the answer will be missense mutation. Could you guys understand this point?
Ma'am, could you please repeat the questions you asked from 1 to 10? Because we, most of the people have missed because we had to attend the other session with other TA.
Okay. So I'll repeat. Let me finish this 14th question. I'll go
Sure.
back. After 14th, I'll go back 1 to 10. Okay. So for this question, could you guys understand why the answer would be missense mutation? So for the
Yes,
frame,
ma'am.
correct, no? Okay, fine. So as per the request, I'll just go back. So just quickly have a look up at these questions. The question 1 is based on the base pairing rule in the DNA, right? That adenine pairs with thymine in DNA and adenine pairs with uracil in RNA. Okay. So using that concept, the answer here will be C. Could everyone understand that? So adenine cannot base pair with adenine. A purine has to base pair with pyridine. Okay, pyrimidine. So purine here is adenine and guanine, and pyrimidine here is thymine, uracil, and cytosine. Okay, so adenine has to pair with the pyrimidine. So in this case, it is uracil for RNA. But for DNA, uracil is not there. It is thymine instead. So the answer is uracil and thymine. Answer is C. Option number C is the answer. Now for this question, those who have like missed it, I would request, I will just erase the answer for you so that you guys can work it out. A newly synthesized RNA has the following sequence. The sequence is given here. You have to determine what is the correct sequence of the DNA template and from which this RNA sequence has been obtained. Those who have like just joined and couldn't look at this question, I request you all to have a pen and paper and try to solve it yourself. So let me do it for you guys. Okay. So this is the RNA sequence. If we write as DNA sequence, we can write it as this just by replacing uracil with thymine. So this will be the sequence of the other DNA strand, the non-template DNA strand, right? One that is running from 5 prime to 3 prime, but the template is will run from 3 prime to 5 prime. So if and this will be the template. We'll use the complementary base pairing rule. T A G G A C C A T. Now in the option, they want us to write in the direction 5 prime to 3 prime. So we have to just read from this end. So it will be T A C C A G G A T. This is the answer, and it matches with the option B. Could everyone do it? Shall I move on?
Yes, ma'am.
Yeah, okay. So for this question, please have a read. It's very easy actually. They are asking here that in the same individual, these are the following cells that are present, and which of these will have a different chromosome number. Okay, so the liver cell, the neuron, the white blood cells, these are what? These are present in different kind of tissues in the body, right? And they play different kind of functions, right? So these are collectively called as somatic cells because they have 2n chromosome number 46. But the cells which are produced in the gonads, like in males it is testis and females it is ovary, which are ultimately going to involved in the process of reproduction. So there the chromosome number has become halved because of the process of meiosis. So it is the ovum which is now going to have N, but this cell is present in your body only, okay? But all of these cells have 2N. So the answer here will be ovum, because it is the only cell which has haploid number of chromosome numbers, like 23 chromosomes only. Rest has 46 chromosomes. Okay. The next question. The next question says the region of the chromosome that joins a pair of sister chromatids is known as what? Means if you recall, this is the structure of a condensed chromosome. Correct? This is the structure of the chromosome that you can see during the metaphase stage, right? In mitosis, there are 4 stages. One of the stages is metaphase, right? So all the DNA has condensed so much that it has taken the form of this structure. This is the chromosome. So they're asking, and these two are the sister chromatids, sisters. And this sister chromatid and this sister chromatid are the exact copies, and these are exact copies, because at the after metaphase, what happens from this location, these two sister chromatids get separated in opposite directions. Correct? So what is this called? And then they are asking, what is this region called? So the center is called the centromere. Okay, where the spindle fibers are connected, and then during means anaphase, they separate out after metaphase. Okay, and from this region, these are the telomeres, which protects this DNA chromosome structure. Okay, these have the protective roles, and entire chromosome has DNA in it, and is different. It corresponds to different regions, that codes for either genes or it is a non-coding sequence, anything. So this is the answer is centromere here. Means centromere and telomere. The answer is D. Now in this question, what they're asking is, what is central dogma of molecular biology? It basically says that how the genetic information is actually flowing within a biological system. And this central dogma of molecular biology is universal. It is same for all organisms, okay, all living organisms. So DNA, which actually contains the genetic information, some part of the DNA, we know a gene, right? A gene is a section of the DNA. It gets transcribed into RNA, okay? And when then RNA gets translated, RNA has genetic codons, which corresponds to each of the amino acids. So each of the amino acids are being read, okay, one by the machinery, protein synthesis machinery, and forms and then arranges the amino acids in a particular sequence, so that ultimately the polypeptide we get, okay, the polypeptide chain we get, and this polypeptide chain is the protein. So the polypeptide chain itself can function, or it can function when it associates with another polypeptide chain, or like more than one polypeptide chain it can associate, and then it can function. So the protein is the functional unit, like everything in our body, like every biochemical reaction and everything, some kind of protein is involved, either in form of enzyme or hormone or like anything, okay. There's receptors, ligands, etc. So this is the central dogma of molecular biology. So based on this understanding, we have to select which one of these the correct option, okay. So where the process happens, like where the replication happens, where the transcription happens, okay. So we know replication and transcription, okay, it happens in the nucleus, all right. But once the mRNA is formed in the nucleus, it is transported in the cytoplasm, where the translation process happens, where the ribosomes, okay, the ribosomes are present and like facilitate the process of translation to form the protein. So among these options, I discussed previously that option D is correct. Why? Replication, it happens in nucleus, fine, but replication doesn't involve copying DNA into RNA. This option is not correct. Then transcription doesn't happen in cytoplasm, it happens in the nucleus. Okay, so this option is automatically wrong. Then transcription, yes, it happens in nucleus, but it doesn't involve copying RNA into protein. So option C is also wrong. The correct option is D, because translation happens in cytoplasm and it involves copying RNA into protein. All right. Next question. The essential components are excised off from the pre-mRNA strand with the help of splicing factors to generate the mature mRNA. Identify the component which is not part of the mature mRNA strand and that must be translated. All right. So here they're asking what part of the pre-mRNA is not retained in the mature mRNA. So the answer is intron here, as you recall from the video lectures, right? So in the pre-mRNA, there is exons just separated by sequences of introns. Okay, so we say that introns are non-coding. Okay, so it doesn't become part of the final protein. Okay, polypeptide chain, but the exons become part. And the UTR, which is present both at the 5' and the 3' end, are also exons. And after the pre-mRNA is processed, we in the mature mRNA, what is there at the 5' end? There is a cap, correct? And at 3' end, there is a poly A tail. So among this option, it is only the intron which is not present in the mature mRNA. All the three exons, UTRs, okay, poly A tail, 5' cap, all are present in the mature mRNA. Now, during the process of splicing, which is not required. What is not required for the process of splicing? So, if you recall, what is the process of splicing? It is the process through which the introns, introns which are present in between adjacent exons, these are removed. Okay, these are precisely cut. Okay, double strand breaks are made and then precisely cut off, and the two adjacent exons are then ligated together. Okay, so for that there is a complex called spliceosome. So the spliceosome is composed of this small nuclear ribonucleoproteins. It is composed of this, as well as it has other proteins involved. Okay, so these other proteins will help in double strand break and then ligation. Okay, so this spliceosome has to recognize where to sit on the intron. Like there is specific sites, DNA sequence sites that spliceosome can recognize and sit. Okay, make the cut and remove the intron, and the remaining exon sequences are then ligated. And yes, for obviously for splicing to happen, we need the pre-mRNA molecules. But what we don't need is the ribosomes. Ribosomes are not part of the transcription process at all, so it won't be in playing any role in splicing. So answer will be B here. Now in this question, what they're saying is protein isoforms. What are protein isoforms? These are the proteins which are coded by the same gene, and they are comparable to each other, and they perform similar roles inside a eukaryotic cell. Which of the following process lead to the generation of such protein isoforms? Right. So the here answer is alternative splicing. So it won't be any like splicing. Simple splicing just leads to if this is a pre-mRNA, simple splicing will just ligate 1, then 2, then 3. Okay, that's the only combination of splicing that combination of exons that we'll get in the mature mRNA. But in alternative splicing, what happens? The sequence of exons is changed. Okay, so here like exon 1 is adjacent to exon 3. Okay, so a 2 can be adjacent to 3, 1 can be adjacent to 3. Like in this way, means the sequence in which the exons are arranged in the final mRNA is changed, right? And in the lecture, sir has explained like what are the ways that alternative splicing can happen. It's a very complex process, all right? Um, and also gene duplication is not in the option because the same gene is being duplicated. There is alternative translation, and this refers to the translation process. Okay, the amino acid sequence becomes alternative. So the option here, the correct option will be alternative splicing, which is actually giving rise to different uh, different mRNAs, mature mRNAs, which only varies in the sequence of exons that they have. Okay, but they all come from the same DNA sequence. And when this mature mRNA is translated, they form different proteins. The function of the proteins, the what function they play, they are similar, but they are not same. Okay, similar. They do similar function, but they are not same. So this has to be remembered. And these proteins are thus called protein isoforms. All right. And this protein isoform is coming from the process of alternative splicing. So the answer here will be B. Now in this question, mRNA is given, which is having 120 nucleotides, right? And when it is translated, it forms a polypeptide chain. And they want to know how long this polypeptide chain will be. All right, and you have to make an assumption that the stop codon also codes for an amino acid. Okay, so it's quite easy, right? So one codon consists of, it's a three alphabetical letter sequence, right? So this is ABC is one codon, and this one codon codes for any amino acid, any one amino acid. So using that logic, if there are like 120 such letters, or 120 such nucleotides, or 120 such bases, okay, and when we divide it by 3, we'll get what, how many we'll get the number of amino acids it can this sequence is coding for. So using this logic, we can calculate the how many number of amino acids will be there in the final polypeptide chain. So the answer will be D. Next, for this question, I request you all to try it out. So those who have like joined late, can you guys see the chat box? In the chat box, I'm sharing a link, okay, a website actually, where you can find the list of all the codons, all the genetic codons, what each of the genetic codons codes for, like the name of the amino acid. Then the amino acid is usually represented using a 3-letter word like um, leucine. You have heard of this amino acid, right? Leucine. This leucine is often written as Leu, a 3-letter code, or it is written as simply L. All right. Similarly, there is methionine. It is often written as 3-letter form as MET or M. So similarly, you can find for all the amino acids. So in this website, you can go and have a look, all right, and then deduce this sequence, like understand this sequence, right? So here the sequence of the protein sequence is given in the form of 1-letter code for the amino acid. Okay, please have a look. Okay, and then try it out. So what you have to do, you have to first get the amino acids, what M stands for, what L stands for, what I stands for, okay, which amino acid it stands for, I'm trying to say that. Okay, and then for that amino acid, you have to get the codon and arrange it. That's all. Arrange it in the same sequence that is given. Okay. Once you arrange it, remember you're identifying the sequence of an mRNA. So for mRNA, T is replaced by U. Okay. So you have to take care of that, and then you will have your answer. It is that easy. Okay. So let me those who are like done, they can just match the answer while I'll solve, or like who is not able to do, they can do it with me. Okay. So here M stands for methionine, and methionine is often and methionine is the start codon. Okay. Wherever the start codon is present, from there onwards the protein synthesis begins. So it is written with it is represented by the codon AUG or ATG. Okay. So in the website they have written ATG. I'm just replacing T with U for mRNA. Okay. Then there is L stands for, here L stands for leucine. Okay. Now leucine can be represented with by two codons, TTA and TTG. So this kind of a behavior of the codons or characteristic of this codon is called degeneracy. Okay, so one amino acid is represented by multiple codons, more than one codons, but one codon only codes for one amino acid. Like TTA, it cannot code for serine. Okay, it only codes for leucine, but leucine can be uh, represented with two codons. This behavior is called degeneracy. This is a characteristic of a genetic codon. Okay, so this is for leucine. Like I'm just arranging. Okay, then there is isoleucine. I stands for isoleucine. Three-letter code is ILE. So it is given by actually three codons, right? ATT, ATC, and ATA. Correct? Here I should write T if I'm writing T everywhere. Okay, this is for isoleucine. Then there is L, stands for leucine, correct? And then Y, letter Y, actually stands for the amino acids tyrosine, TYR. It is written as TYR and is given by codon, these two codons. Okay. Then you have to just check the options which matches with this sequence. This is 5 prime to 3 prime only. Okay. So translation happens in 5 prime to 3 prime region, and you have to find the mRNA sequence in 5 prime to 3 prime region. So this sequence is already in 5 prime to 3 prime. You don't have to worry about that. Thank you for muting. Okay. So here let's match the options here. ATG, that stands for AUG. Here also it is present. Here also it is present, correct? Then TTA means basically UUA. You can write it as UUA. So it stands here, here, here as well. But here AUG was not present. So C is gone. Like C is not the correct option. Okay. And here also UUA is not present. The B is also not the correct option. It is between A and D, correct? Then this is for the isoleucine. You have to match ATT. So AUU. So AU is present here as well, present here as well. Then this is for, again, leucine is coming. Leucine is given by both UUA as well as UUG. So instead of U here, it was UUA, but here it is UUG. So the meaning remains the same, but different codon is present. So this is option is correct. Here UUG is not there. So this is not the correct option. This is the correct option. As we'll see that UAU, that is TAT, is present. This is coding for tyrosine. Tyrosine, and then this is the stop codon. Stop codon will be present. Okay. What is the role of stop codon? It actually tells the machinery, protein synthesis machinery, to stop here, but it doesn't actually code for any amino acid itself. So whatever polypeptide sequence that you'll get will be before the stop codon. Okay. So stop codon will be there. So this is your answer. Option A is your answer. Is everyone like, could everyone understand this? Okay. No keep. So like, I think I can continue from here, or I should like go like from question number 14. From question number 11, could everyone was there, like, um, understand the questions from question number 11 onwards? Yeah, I
Yes,
see
you
a
can
thumbs
go
up.
ahead.
Yeah. Okay. So previously I stopped at question number 14. Now I'll move on. There are a few more questions left. Those who have I'm seeing many people are joining late actually, so they can actually, tell me. I can go back and touch upon few questions that they have missed. Okay, so in this way everyone is on the same page. Yes, Amardeep, you have a question? Amardeep,
No,
you
ma'am,
have.
just
Okay, actually you raised your hand, that's why. Okay.
mistake.
No problem. So in this question, it is upon the concept of karyotyping. Can anyone tell me what they mean by the term karyotyping? What is karyotyping? What this term means to you? Is everyone has seen the week 1 lectures, the 4 lectures that Professor Ganesh have uploaded in the NPTEL platform? So if you are familiar with those lecture content, you can definitely say,
Yes.
yeah. So what do you mean by karyotyping?
A diagrammatic representation of all chromosomes.
So it is not a diagram. Actually, you don't draw diagrams of the chromosomes. So you actually, you know,
Pictorial presentation. It would be a pictorial presentation.
so it is actually an image. Okay, it is the image of the chromosome. We are not making cartoons or anything. We take cell, okay, any somatic cell you can take from an individual, like from blood or anything. So and then you
Arrangement of
obs
chromosomes based on size and shape.
correct. So you are correct. So we are actually trying to see what is the shape, okay, what is the size and shape of the chromosome that is present inside a cell, so so for doing that we have to first collect the cells from the body, like somatic cell, any type of somatic cell you take, okay, and then you have to see in the metaphase state of the cell, when the cell is in the dividing, like when cell is in the cell division process, because at the metaphase the DNA is actually condensed to form chromosome. If you observe cell in any other like normal phase, like when it is not in the cell cycle, you cannot see the chromosomes. So you have to observe the chromosome when the cell is in metaphase state. When you have confirmed that the cell is in metaphase, you have to then go ahead and fix it, okay, and then get image of, you use electron microscopy, okay, and then you get the image of the chromosomes, how they are looking, okay, and then you see the different sizes. So chromosome 1 is the longest chromosome, whereas chromosome 22 is the shortest. In this way, you cut the images and then qualitatively understand the shape. So, and whatever arrangement in the you will get for different chromosomes is the karyotype of that individual. From that karyotype, you can say the sex of the person, or any chromosomal abnormalities are happened or not for that individual. So so karyotype is very useful. Okay, so in this question they are asking that you are going to make karyotyping from blood cells. So blood cell is like it's a somatic tissue, right? So it will have not all the blood cells. They are going to ask like which of the blood cell you are going to use for the karyotype. So they're taking blood from the body and then visualizing the chromosomes under the microscope to see whether there is metaphase or not. And then they have separated the population. There are different population of the blood cells, right? The red blood cells, white blood cells, platelets, etc. And then you are asked that which type of blood cell would you choose in order to prepare such a karyotype so that you can do your further study. So what do you think will be the answer?
Ma'am, WBC.
WBC. Okay, so why do you think WBC will be chosen?
It contains nucleus.
Correct. Why not platelets?
And they do not contain nucleus.
Okay, they do not contain nucleus. Actually, what do you mean by platelets? What are platelets actually? I'm not very much convinced. Like they do not contain nucleus means, uh, it doesn't give a clear picture. So how it is then different from RBC? If anyone asks you, would anyone like to think and try to give an answer on this? So the correct answer is the white blood cells, right? Definitely, whoever gave the answer, you are correct. But why not? Why can't we choose platelets then? It's obvious. You guys have studied in your class 12 level that red blood cells are enucleated. It doesn't contain nucleus at all. So red blood cells definitely can't be used for karyotyping. But why not choose platelets?
Ma'am, because platelets are basically involved with the means the clotting of the blood. If any cut happens, then platelets forms a clot on the cut, ma'am, on the cut part, ma'am.
Platelets actually secrete the clotting factors, and those are then involved in the clotting process, the wound healing process. You're correct. Please, I request you all to have a clear idea like what are platelets, okay? But the answer here is white blood cells. Means what are actually platelets? How do they look like? Please do have a Google search later on after this class, because I feel like this you guys should know, okay? And you guys should figure it out yourself, okay? Now in this question, here you are given a karyotype, okay, where two chromosomes are shown, chromosome 9 and chromosome 12. You have to examine this photograph and then identify the cause listed incorrectly for the defect. Would anyone would like to try? What would be the correct answer here? What is not happening means whatever this image is showing here, okay, uh, you should understand what is this image showing first. If you understand that, then you can definitely understand what it is not showing. Means you have to that is the correct answer in this case.
Ma'am, non-disjunction.
Okay, so what do you mean by non-disjunction? The term non-disjunction, what do you mean by that?
Some chromosomes do not separate at the time of meiosis.
Okay. So understand in this way. So at the end of meiosis 2, like there's meiosis 1, meiosis 2, right? So at the end, we get like from one mother cell, we get 4 daughter cells, and each have haploid, right? From 2n, the chromosome number becomes n in this case. So while it is happening, the sorting of the chromosomes is not happening equally. Like if this is the case in mother cell, okay, a daughter cell should, so there is 1 2 1 2 3 4. This is one like one pair of homologous chromosome that is coming from mother, means another is from father, right? Like this person has received these 2 homologous chromosomes from their parents. Now she is going to pass it on to her, next generation. So here 1 is coming, 2 is coming, 3 4. So what I'm trying to say, the equal distribution is happening of the of each chromosome. All right. But in non-disjunction, what happens? The sorting is not equal. The sorting is not equal. Means if this is daughter cell 1, 2, 3, and 4, one has received 1 chromosome. Okay. Now 2 has received 2 chromosomes, but 3 has received none, and 4 has received 1. So the cell that is the third daughter cell is actually missing one entire whole chromosome, because it got sorted into daughter cell number 2. So this daughter cell number 3 is devoid of one whole chromosome. So when this type of thing happens, the number of chromosomes changes. So for this person, whoever is going to is whoever the offspring is going to receive this gamete, which is having which doesn't have the chromosome, one chromosome, means the final chromosome number in that person will be 45, right? Whereas if the person who receives this gamete, her or his chromosome number will be 47, because that person is receiving one whole extra chromosome. This is non-disjunction. This is happening because of the non-disjunction, which results in increase or decrease in the chromosome number, or change in the chromosome number in the individual. Okay, so here in this figure, the chromosome number is not the case. So the incorrect so the answer here will be obviously non-disjunction. But what is happening is translocation. All right, what is translocation? Translocation is the thing that happens due to the recombination. If you remember the term recombination, that happens during the meiosis 2, again meiosis 2 stage. We divide meiosis 1 and meiosis 2. Meiosis divided in this case, right? So during recombination, some part of parental homologous chromosome is transferred to maternal one, and vice versa. So this kind of recombination is happened, which leads to variations, right? Variation in the offspring, some offspring. So now if this translation happens, for the region of the chromosome that is not needed, and the resulting gene that is being formed is uh, unfortunately becomes like defective. It leads to a defective uh, protein, or the translocation happens in such a way that 2 genes comes in contact which are not supposed to, like in normal condition, and they start expressing together, and the resultant protein again is causing a disease or something like that. So this kind of condition then causes disease. So so this is a classic like where chromosome the translocation happens between chromosome number 9 and chromosome number 12. It leads to the formation of Philadelphia chromosome. You must have heard this term in the lecture. So in Philadelphia, chromosome is formed and associated with the type of a leukemia. That's a cancer, blood cancer. All right. So in Philadelphia chromosome, two genes, BCL and ABL, they come in contact, which are not supposed to. Okay, which are not supposed to come in contact in normal, and then they start expressing together and leads to abnormal protein. Okay, and start expressing that abnormal protein and leads to cancer. So this example is related to that condition that is discussed in the lectures. So for this question, the correct answer is non-disjunction, because this image is not talking about non-disjunction. It is rather talking about translocation, and translocation is happening because the double strand break or the chromosomal break. Okay. Now have a read at this, have a look at this question. Which one of the following options correctly matches the syndrome to the respective karyotype? Okay, so just before I was talking about non-disjunction, where the number of chromosome either increases or decreases. So if you remember, if you recall from the lectures, sir has discussed three kinds of syndromes. All right, that results because of this non-disjunction and then change in the chromosome number. One is Down syndrome, right? Turner syndrome, and then Klinefelter syndrome. In Down syndrome, what happens? The individual who is having Down syndrome has 3 copies of chromosome 21. Okay, so if we represent that person's chromosomes, it will be then 47, और ये तुम्हारा एक extra 21 chromosome होगा उसमें, right? That person has one extra copy of chromosome, extra copy of 21 number chromosome, right? So for this option A, it is not the case. Down syndrome has this type of a representation of the number of chromosome. It is not this. So option A is not correct. For Turner syndrome, if you recall, the person is actually lacking one X chromosome, all right, or Y chromosome, and that person unfortunately has only one X chromosome. So for Turner syndrome, the person has 45 at X0. That person's chromosome number can be represented like this because that person is missing one copy of the sex chromosome. So for Turner syndrome, again, this is not the correct representation. Now, what about the Klinefelter syndrome? This Klinefelter syndrome person, whoever is having the syndrome, will have one extra copy of X or Y chromosome, okay, coming from the mother or father. So the mother is passing on the gamete and the ovum, and instead of having one X, it is having two X, all right, and the father is giving one Y. So then the person resulted with this kind of a chromosome combination, right? It is having one extra sex chromosome, either X or either Y, you can say. So this is the correct option here. All right, and this is obviously not the correct answer. So among these four, this is the correct answer, where the Klinefelter syndrome person will have one extra copy of the sex chromosome, either X or either Y. Now in this question, again, it is based on the karyotyping. Given below is the karyotype of a person suffering from a congenital hernia. Then we have to examine and identify these options, like which of these options actually means from this karyotype we have to understand which of these options are correct. Okay, so this arrow shows that we have to focus on chromosome number 15, like one of the homologous chromosomes. Okay, we have to focus on this. Now now whoever the karyotype of whoever is this person is male, correct? is having one pair of X and one pair of Y. So and also all pairs like 1 to 22 pairs are present. So we can write as 46XY for this person first. Okay, so 46XY is present here, so is present here as well. So this option is wrong, like you can understand why it's wrong. Okay. Now here at chromosome number 15, observe like this chromosome, the structure of this chromosome is ring like, means circular. If you remember, like sir has explained in his lecture, like what kind of chromosomal translocations can happen, like, and how it can happen, whether there is break in one chromosome, right, one break in one chromosome, two break in one chromosome, or two break in two separate chromosomes. Okay, so because of that, how the chromosomal structure is changed. So there was one case where this is the normal chromosome. Okay, and in this normal chromosome, break is happening at two locations. Okay, so two breaks in one chromosome, because of which the broken ends, this is the centromere, this is centromere, they form a ring-like structure. So similar to that example, we see this ring-like structure. Okay, and this part is destroyed, like it is deleted, correct? So this ring-like structure is coming from deletion, right? Deletion of few segment of the chromosome. So inversion is not happening. What is inversion? So if this is the chromosome and this is the segment of chromosome in question, and normally it is present as AB, right? Means this is the normal orientation of this sequence. Because of double strand break at this A and B location, the sequence has now inverted to BA in the final chromosome. This is the inversion. This is what is called inversion. So inversion is not happening. Inversion is not giving me a ring-like structure. Okay. Translocation. Translocation, as I told you in the Philadelphia chromosome example, right? So two chromosome has to be involved. We need two chromosomes first of all, and there will be double strand break in these two chromosomes, and these two chromosomes can be doesn't necessarily have to be homologous, they can be non-homologous chromosomes as well. And then a translocation, like this sequence will go here, this sequence will grow here, right? So this kind of translocation can happen. So but that again doesn't give the ring-like structure. So the correct answer is A, right, which is giving the ring-like structure. Could everyone understand this example? So from the karyotype, we are here understanding like what is the karyotype. The karyotype is given, and from this karyotype, from this given karyotype, we are trying to answer the reason, the cause of the defect. We are trying to answer that, as well as we can determine the sex of the person, whoever the person's karyotype is. So with this, I have like completed all the 18 questions, which are related to the week 1 content given to you. Okay, and I hope like whatever questions that I have solved today will help you, go back and have a look at the assignment problems which are given to you guys, and you can understand those assignment problems better. Okay, so if anyone has any query on the week 1 content, or assignment problem that is given to you, or if anyone has missed any certain question, any one of the 18 question, and they want me to go back and explain, please let me know.
5, ma'am.
Yeah, yes ma'am. Yes, bataiye. What is your query?
1 to 5 questions, ma'am. Can we go quick revise?
Okay. So you have joined late, right?
Yeah, ma'am.
Oh, okay, no problem. So this is question number 1. Here, this question is based on the base pairing rule, Chargaff's base pairing rule, which says that in DNA, adenine will always base pair with thymine, correct? Whereas in RNA, adenine will always base pair with uracil. So using that concept, we can understand from the option that option C will be correct, right? Here it will be uracil, adenine base pairs with uracil in RNA, and it base pairs with thymine in DNA. All right. Now in question 2, fortunately the pen is not working. Okay, fine. So in this question, you are given mRNA sequence, okay, and from that you have to deduce the sequence, the DNA template. Okay, so you can work it out yourself, or I'm just quickly showing you. The answer is given here. So this is the mRNA sequence. I've just converted into the DNA sequence by changing uracil with thymine. All right, and then with using the complementary base pairing rule, I have obtained the DNA sequence of the template. This is the DNA sequence of the template. All right, this one by change the direction. All right, and since the option in the option we have to write the sequence in the direction 5' to 3', so you have to write starting from 5' T A C, like in this way you can determine that answer. Option number B is the answer here. Now next question. Now this question asks you, like, among one of like among these 4 cells, which among these 4 cell types, which one is having a different chromosome number. All right, so the liver cells, neurons, the white blood cells, these are all somatic in nature, right? So they are present in different kind of tissues, right? And different playing different roles. But it is the ovum which is the gamete, the term gamete. So they are haploid in nature, so which is why it is going to have a different chromosome number, because ultimately it is going to fuse with sperm and then form an zygote. So the whole fertilization process will take place, right? So it is in the body, it is the ovum or the sperm which is having a different chromosome number, and where the rest of the cells in the body all are 2n. Okay, all are having 46 pairs, the 46 chromosomes. Only ovum has 26 chromosomes. Now in this question, you have to understand the structure of the chromosome actually, which is formed during mitosis or meiosis, when the cell is going to divide, during cell division. The region of the chromosome that joins a pair of sister chromatids is known as. So here, this is the structure of the chromosome that I have drawn, and these two are called sister chromatids. Okay, and on one side, the sister chromatids, they're identical. Okay, so this is identical to this. This region is identical to this. Okay, and these two sister chromatids are connected by the centromere. Okay, centromere, and the extreme ends of the chromosome here and here, these are called the telomeres. So the correct option is D. Now, question number 5. This is based on the knowledge of the central dogma of molecular biology, which shows the flow of the genetic information, and it is universal. It happens in all organisms, all living organisms. The DNA, which actually contains the genetic information, and is it is passed on. We pass on to the next generation, or we get it from our parents, right? So, few segments of the DNA, which is gene, right? It is transcribed into mRNA, correct? So from mRNA, the protein synthesis machinery translates it, okay, and form the protein. So protein is nothing but a sequence of amino acids are joined together by peptide bond and forms a polypeptide chain. So, and also the location of the transcription, translation, these varies. All right. So replication, where copying of DNA into DNA only, right? That happens during cell division. That happens in nucleus. Transcription, formation of RNA, mRNA. This happens in nucleus, but the mRNA, once it forms, it goes ahead and in the cytoplasm, where there is ribosomes, and that facilitates the process of translation. So using this knowledge, we have to look at the option. Replication, it happens in nucleus, correct, but it doesn't involve copying DNA into RNA. Then transcription, it doesn't happen in cytoplasm. All right, this option is also wrong. Then transcription, yes, it happens in nucleus, but it doesn't involve copying RNA into protein. So the correct answer is D. Okay, but where translation is happening in cytoplasm, and it does involves reading the genetic codons in the mRNA and then translating it into a sequence of amino acids. All right. So if anyone would like to, I'm done with explaining 1 to 5 questions. If anyone else have any other doubt other than these questions, which is based on the week 1 content, or would want me to explain any of the questions again, please let me know.
Yeah, could you please continue if the time is permits you with the second week lectures or few topics from the second week?
So like I'll touch upon, like I'll make dedicated slides on second week topics, and then in the
But
next
if you
week.
can solve few questions from second week, it would be great. If you can do, otherwise it will be fine. Left for next week or second time, the next time.
I think deadline for second week assignment is also 8th, right? Or it is
Yeah.
it's also 8. Okay, so that's why you want. I understand. Okay. So right now I have not actually prepared for second week, but it will be discussed in the next week only. I'm just focusing on week 1 content right now.
Okay.
Ma'am, what is truncated protein?
So the word truncated means shorted. Okay, shortened. So truncated protein means um, if I get a blank slide. So if this is here, this line say represents polypeptide chain. Like randomly, these are some amino acids. All right. Now, due to some mutation, right? Say nonsense mutation, okay, or frameshift mutation, you get premature. Like here, they're supposed to be stop codon. So stop codon in the mRNA. So that resulted in the polypeptide chain of this much length. But due to like mutation, like nonsense mutation or frameshift mutation, you get a stop codon upstream, like before it is intended, say somewhere here in the mRNA. Okay, in the mRNA, because of which the final protein length has become shorter. Previously, it was of about 10 amino acids. Now it has become only 4. Right, and so the protein, the length of the polypeptide chain, or the length of the protein, the original length has become shorter because of this mutation that has happened in the DNA sequence, and it is then read in the mRNA and then translated into the protein. Is that clear?
Yes, ma'am. Thank you.
Okay. If anyone else have any query, please let me know.
There was one question in the assignment that which statement is not true about the imprinting disorder. And ma'am,
Okay.
option
Yeah, option.
options were ma'am, they are caused by loss of function of mutation. Second option is defect non-coding repetitive sequence. Third is epigenetic mechanism is involved. And fourth is shows sex-specific inheritance.
Okay. So see, I cannot tell you the answer. Okay. um, so I will just explain like what are imprinting disorders. Okay. From that you have to figure out what will be the correct option, because after the submission of the assignment you guys will definitely get the solution. Okay, so from that you can match the answer, or like the next week, by next week also you will the submission deadline will be over. Then if this question you think, if you have still doubt in this question, then I'll again explain each option individually. For now I will just explain what are imprinting disorders. Okay, so imprinting disorders actually refers to a special category of disorders which are related to imprinting genes, imprinted genes. So what are imprinted genes? Now there are few genes, okay, in the body uh, which are only expressed if it comes from either father or from mother. So if there is gene 1, if it is coming from your mother in your body, okay, then it will be expressed. But if this gene is contributed by your father, then gene expression will not be there. Okay, so these genes are imprinted. uh, this is called imprinted genes, means uh, these genes has to come from their parent, like one of the parent, for them to be expressed. So this is the pair of homologous chromosome. Now gene 1 present here as well, here as well, and this copy is coming from your mother, this copy is coming from your father. Okay, now in normal case the father is also have the father the chromosome that's coming from the father is also having gene 1, but it is either deleted in normal case, or if it is present it is silenced. So there are various silencing can be done by various non-coding mRNAs. Okay, so they can silence the this copy of the gene from the father, the chromosome coming from the father. Only this is expressed. Now, in the disorders where imprinting disorders, what happens? This case happens means the gene that was supposed to come from the mother and cause expression, it is not happening. Okay, disorder will happen when this type of expression is not happening. Means mother is supposed to give the gene 1 and then cause the expression of the gene, but due to mutation the copy of the gene that is coming from the mother is also mutated and lost its function. So the person is now having defective gene 1, even if the gene 1 is present in father, but it is silenced, so the gene is not at all expressed in that person. So there are actually diseases, like example I can give is Prader-Willi disease, Prader-Willi syndrome actually. Then there is Angelman syndrome. And these are like related syndrome. There is one region in chromosome number 15. Okay, and if the and if in Angelman syndrome, what happens if this chromosome number 15 is not coming from the mother? Okay, the paternal chromosome number 15 region in the paternal one is silenced or deleted. Okay, and if it is not coming from the mother, this syndrome happens. All right, but the ulta, like the opposite is true for Prader-Willi. This region of the chromosome number 15 has to come from the father, or the chromosome copy from the father. If it is not being expressed from the father's chromosome, this region, then the Prader-Willi syndrome happens. So this is like imprinted genes and imprinting disorders. Actually, this thing will be discussed in detail, I think, in subsequent week 1, week 2, where maybe sir like to talk more about Prader-Willi syndrome and how these imprinted genes are inherited through generation, like the pattern of inheritance. So could I explain, like, could you understand what are imprinted genes and how the imprinted disorder
What
is happening?
I might have. Can it be inherited in both male and female offspring from the parents in case of imprinted disorders?
So this thing, you will understand better when when sir explains that tree, family tree, and how the disorders are being inherited. So there you will able to understand much better. So this question which I've asked is not covered in week 1 content. Okay.
Means the expression of the genes which are being inherited from the parents. You said one is like silenced, other is expressed, whoever it may be, whether father or mother. So in case of inheritance, inheritance would be there, right? Half inheritance, inactivated even, or one is expressed. So how it is expected to be expressed? Is it going to be expressed in both? Is there any chance, or is it gender specific, or it is gender? I think neutral. It can be inherited to any male or female child.
So both male and female will have one copy of mother and father, right? Mother and father chromosome, correct?
The disorder, the imprinted disorder, like that could be inherited in both child, or it is specific. Suppose if mother is inherited, it can only go to the female child, or if the father is inherited, it can only go to the sons or the son.
I think you're asking me whether that option is correct or not. That option given in the fourth one, right? Actually, we are told not to cover the assignment question, current assignment questions. So if I tell you yes or no, then you'll definitely have a clue, right? But it is up to you to figure that out. Maybe if you're familiar with how that inheritance family tree, if you're familiar with it, like how the different kind of disorders or genes are transferred, from father to mother, then from grandfather to their grandchildren. If you're familiar with that concept, you can work it out yourself. But I'm sorry, I cannot answer to this question.
Can understand. You
Okay.
can
Yeah.
but
Okay.
it like is included in the assignment questions, but in thinking of a broader aspect and thinking about of the overall concept of this imprinted disorder, it would be incomplete if we don't know this.
Okay. Okay. So, as far as I have the knowledge on this topic, I feel like uh, the XX, like if the disorder is related to the sex chromosome, okay. So, if the gene that is in related to a sex chromosome, then the sex specificity will come. Now, think in that direction. Mother will only contribute to X chromosomes, right? So imprinted disorders or the imprinted gene can be located in any of the chromosomes, all right? But the sex specificity usually comes when it is associated with the X and Y chromosome. So this might give you a clue on that question. All right. Could you understand the point that I'm trying to make? Sex
Can you repeat
specificity.
it?
The sex specificity will come when your when your means defective gene is present in the sex chromosomes, the X or the Y chromosomes. So if any defective gene that is contributing to a disorder is present in Y chromosome, that Y chromosome is transferred from father to his son. So that sex specificity is, understood, right? Even in case of mother as well. So
Is it genetic mutation or like says it is a mutation present in the allosomes?
so see, imprinted disorders can be present in allosomes, sex chromosomes, both, right? The genes, imprinted genes can be present. Okay. But the sex specificity thing, how it will come, I'm trying to give you a clue. See, I cannot give you the answer. So I'm trying to give you a hint, like what is actually is. Like, say, epigenetics is the option, right? So that epigenetics is coming from the environment. The environment is causing some change in the DNA, which may not be permanent, like which may not be completely changing the DNA sequence. It might be reversible as well, and it may not get passed to the to its future generation. You think of an answer for now. It's just 1 point question, and next week I'll like explain all the 4 points that is given here on imprinting disorders in more detail. What is right according to you? Just answer for now. Okay.
Sure.
Yeah, sure. Okay. Like, actually, imprinted disorders, it's a very big topic. Many research papers are published in this area. Even when I was doing this course myself, I had to read many research articles and all. So maybe in next class I'll cover those topics with you, and you'll have a more detailed idea of what are imprinted disorders. Okay, which sir has not see, actually in the lecture sir has told you to look into it yourself. So maybe you should do that as well. Look into some research papers and all. You'll have an in-depth idea. But next week I'll definitely discuss those 4 points. Now, if anyone else have any query, um, please let me know based on the week 1 content. Okay. If not, then we can then end today's session. All right.
Which file is shared?
Yeah.
Can we get some time to solve some questions from the second week? Not now, but is there any chance that we can get some time or some session provided this week before its deadline?
I'll ask the NPTEL coordinators. Okay.
Okay.
They will manage it. Okay. I'll ask them if means we
Because
were told
complex
that
to be learned. Many topics are there. Those have to be learned in a precise manner. Without that problem solving session, it would be very mess for us to solve.
okay. So few more sessions are there, right? Monday and Tuesday there is also those session. Maybe I'll ask those TAs to cover few week 2 topics as well.
It would be great.
Okay. Then NPTEL people won't be bothered at all, and those people can cover your doubts based on week 2. So today I think me and another person took the session, and both of us covered week 1 content, isn't it? Yeah. So Monday and Tuesday 2 more sessions are there. I'll ask them, like I'll talk to them once, or I'll talk to NPTEL people as well. Something can be definitely done about it. It's not a big problem. Okay.
Okay, thanks.
Okay. So thank you all for joining. next time I would request a bit more participation from you guys, so that solving these questions would be, fun for all of us. Okay.
Thank you.
Yeah.
Thank
You
you.
guys
Thank
can.
you.
Yeah. You guys can leave the meeting. Thank you for joining.